Hello everyone. I will present the project Signature of Selection, Coevolution, and Co-regulation in the CYP3A and CYP4F genes in humans. The project was done in collaboration with Alex Richard Saint-Hilaire, a previous master student in the laboratory of Julie Hussein, but I would also like to thank Justin, Jean-Christophe, Raphaël, and Julie. The field of pharmacogenomics wants to study the effect of a mutation on the response to a drug. The aim of this field is to personalize treatment based on the patient genetic. An example which is used today in hospitals and in clinics is with the CYP2D6 gene. This gene metabolizes many medications, and mutation in this gene impact its activity level on the metabolization. So depending on the genetic of the patient, the dose of the medication can be either full, half, double, or even you need to find an alternative since it will be toxic for the patient. The CYP2D6 gene is a member of the cytochrome P450 superfamily. This superfamily contains 57 genes and 58 pseudogenes. Their main functions are the metabolism of drugs and the detoxification of the body from xenobiotic molecules, so molecules from the outside. They are important for precision medicine and pharmacogenomics since they metabolize around 75% of common drugs. So the schedule of the presentation is that I will first present population genetics, then expression analysis, and finally phenotypic analysis. In the field of population genetics, we can detect if a mutation is under selective pressure in a population. To be under selective pressure, the mutation needs to have an effect on the reproduction rate and/or on the survival of the individual. You have three types of selective pressure. The first one is the positive selection. In this selection, the allele will confer an advantage to the individual, and so it will increase in frequency. On the other side, you have the negative selection where the allele confers a disadvantage to the individual, and so the allele will disappear of the population. The third one is the balancing selection. You will have at least two alleles conferring an advantage. They will increase in frequency until they get to an equilibrium between both alleles. Overall, all the three selections will affect the mutation around them, allowing us to detect the selective pressure using statistics in population genetics. Our first question was to see if we can detect selective pressure in the CYP superfamily and which one. We started our analysis using Tajima's D statistic. This statistic is not the most recent one, but it's still a good starting point. So the statistic will allow us to identify global genetic patterns in each of the CYP genes. A positive D means an excess of alleles at intermediate frequency, suggesting balancing selection, and a negative D means an excess of rare allele, suggesting directional selection. Here you have the distribution of the D of Tajima across the CYP superfamily in the European population. They are sorted by the mean, and usually almost all of the genes have a mean under 0, and only 10 of them have a mean above 0, and among them there is 5 genes of the CYP4F family, and so this suggests a balancing selection in this family. On the other side of the graph, you have 9 genes with consistent value under 0. And among those 9, we have all 4 genes of the CYP3A family, suggesting directional selection. About the first family. The CYP4F family is a cluster of 6 genes on the chromosome 19. Usually, when you see a cluster of genes of the same family close to each other, this is generated by duplication event. And here, the potential ancestor is the F22, which is at the left side of the cluster. They are not well characterized, and there is few association in the pharmacogenomic field. The Tajimas suggested balancing selection in this cluster, and we want to confirm this analysis with a second statistic, which is the beta score. The beta score detects cluster of allele with similar frequency, which can be generated by balancing selection, as you can see in this graph. So a beta score above a 0 will mean that the loci is under balancing selection. Here you can see the beta score across the CYP4F cluster. Each genes of this cluster are represented by an arrow under the graph. The red line is the 99th percentile of the chromosome, and so it is our significant threshold. And we can see the pattern across all populations, so in Africa, in East Asia, in Europe, and in South Asia. We can see many large beta scores, which are replicated across the population, such as the F12 between the F32 and F8. Even if they are weak, they are still consistent between the populations, so in the F23P and the F11. So this confirms balancing selection in the CYP4F cluster. However, the interest in the pharmacogenomic field for the CYP4F family is not the highest, so we will focus on the second family for the rest of the presentation. The CYP3A family is a cluster of 4 genes and 2 pseudogenes on the chromosome 7. They are also generated by duplication events, with the ancestral one being the A43, which is at the right side of the cluster. They are more studied than the CYP4F family, and there is many association in the pharmacogenomic field. The D of Tajima detected directional selection, meaning either positive or negative selection. We wanted to validate this signal using another statistic, which is the IHS. IHS allows us to detect positive selection based on the linkage disequilibrium pattern around a mutation. The IHS value, so either it is positive or negative, will depend if the ancestral allele is under selective pressure or it is the derived allele. Here you can see the absolute value of IHS above 2, which is the threshold. You can see that there is many signals which seems to differ between population. For example, you can see in the African population that the signals are in the CYP3A5 genes. However, in the East Asia, the signal is mostly between the A51P to A4 genes, and the European population are just next to the A4 genes. One gene has signal from all superpopulation, and this is the genes A43, which is the ancestral one. This confirms positive selection in the CYP3A cluster. Then, if the D of Tajima did not detect balancing selection, we still want to verify with a more precise statistic, which is the beta score. So here you can see the beta score in the cluster of the CYP3A family across the population, and we can see that the beta score are lower than what we saw in the CYP4F family, but we still see one significant signals in the CYP3A43 genes in the African population. So this suggests that the CYP3A cluster have both positive selection and balancing selection in the African population. Many families inside the CYP superfamily has tendency to evolve together in a cluster, like you saw for the CYP3A and CYP4F family. We wonder if there could be links between the genes inside a cluster, and so if there could be coevolution between genes. A coevolution is when at least two genes interact together and evolve together. This can be generated by epistasis interaction, which means it is the nonlinear interaction between at least two mutations and two genes. This means that when you look at one gene individually, it won't have the same effect on a phenotype if you look at these genes in interaction with a second gene. In a previous paper, I discovered a coevolution between two mutations generated by an epistatic interaction using an approach based on linkage disequilibrium. So what is the linkage disequilibrium and how does it allow us to detect coevolution? So the linkage disequilibrium is the correlation between allelic frequency of two distinct mutations. During the meiosis, the recombination event can break the linkage disequilibrium between two mutations. Under neutrality, this is what we should expect. So we have the sequence of the two parents, and we want to study the linkage disequilibrium between three loci, so the locus A, locus B, and locus D. We know that the loci A and B are next to each other. So during the meiosis, we don't expect to see recombination. And so in the children of those individuals, the big A will always be with the big B, and the small a will always be with the small b, and so they are in linkage disequilibrium. However, the locus D is further away, and so during the meiosis, we will expect to see recombination event between the locus B and the locus D. So what we can see here is that half of the time we can see the big A with the big D, and the other half it is with the small d. So it's really random, and this is what we expect without linkage disequilibrium. However, during a coevolution, you will have an interaction between, for example, the locus A with the locus D. The big A needs to be with the big D to confer an advantageous effect, and so in the children we will see the big A more often with the big D than what we expect given the distance. And so this will generate unusual linkage disequilibrium. As I just explained, we will expect to see linkage disequilibrium between mutations who are close to each other. And since the cluster of this family are close to each other, we will expect to see some linkage disequilibrium. To verify if what we see is unexpected, and so potential coevolution and not only caused by the proximity of each other, we generated a null distribution. To do this null distribution, we compute many windows of around 430 kb containing 6 consecutive genes, which are approximately the same condition that what we see for this cluster. We calculate the correlation between mutation, and we plot them in this graph, where the genetic distance is the genetic distance between the two mutations. The black line are the 99th percentile of each distance. And so, as expected, we can see a decrease in the correlation when the distance between the two mutations increase. Here we can see the correlation between all pair of comparison of the genes in the CYP3A family. We can see a high unusual linkage disequilibrium between the A43 genes, with both genes at the outer side of the cluster, so with the AA5 and A7. This was only seen in the African population, and so this could suggest a coevolution between two genes. After finding a coevolution between genes in the CYP3A cluster, we wonder what could be associated with this coevolution. The first intuition was at the gene expression, so could there be a co-regulation between genes in this cluster? To do that, we did an expression quantitative tree loci, so an EQTL analysis, using GTEx database. This database has expression data in 54 tissue for around 800 individuals, and the genotype. Here you have the p-value of the association of each mutation under selective pressure in the CYP3A cluster. The black line is the genome-wide significant threshold. We can only detect one locus which is significant, and this locus is in the CYP3A43 gene, and it is for the CYP3A5 gene, which is at the other side of the cluster. If you remember the results I just showed before, those two genes are under coevolution in the African population. Furthermore, the SNP was significant in the CYP3A43 genes. It's also the one which was under balancing selection in these genes. And so this suggests that the CYP3A43 have a regulatory regulation effect on the CYP3A5 expression. The next step was to see if SNPs under selective pressure were associated with phenotypes. We did a phenome-wide association studies using the UK Biobank. This biobank have phenotypic and genotypic data on more than 500,000 individuals. We did associations for around 700 phenotypes. We didn't find any association for the disease, but we found many associations for continuous data. We mostly see association with the biomarkers, so the plasmatic cells. Here you can see the p-value of the association of SNPs across the CYP3A cluster, which passed our threshold. And you have two types of points. The opaque one are SNPs under selection, and the ghostly one are SNPs who are not under selective pressure. We see many signals, but the most significant is for the high light scatter reticulocyte count, which is an immature red blood cell. The most significant SNPs for this phenotype is under selection in Africa, and the SNPs under balancing selection is also associated with this phenotype. A quick summary is that we first identify selective pressure in many populations on the CYP3A cluster. We identify a balancing selection in the CYP3A43, which seems to regulate CYP3A5 expression. And lastly, we found that the CYP3A cluster is associated with many phenotypes, but mostly with the reticulocyte. We wonder if there could be a link between CYP3A5 expression and those phenotypes. And so we use a Mendelian randomization analysis. A Mendelian randomization analysis wants to verify if there could be a causal relationship between an exposure, which is here the CYP3A5 expression, and an outcome, which is here the phenotype, by comparing the SNP effect of both phenotype. And if there is a correlation between two SNPs effect, this could suggest a causal relationship of expression on the phenotype. We evaluated the causal relationship between all phenotypes showing significant association intercluster, meaning 6 phenotypes, with CYP3A5 expression. We performed 3 tests, meaning IVW, and 2 sensitivity tests, meaning Agar and weight median. I show you the only 2 phenotypes showing significant causal relationship, which are both associated with the reticulocyte. On your x-axis, you can see the SNPs effect on CYP3A5 expression, and on the y-axis, it is the SNPs effect on the outcome. We can see that when there is an increase of CYP3A5 expression, we can also see an increase in the outcome. Overall, our Mendelian randomization shows causal relationship between CYP3A5 expression and the reticulocyte phenotype. We wonder what could be the origin of the coevolution between the gene A5 and A43 in Africa. So in our analysis, we found that this cluster could influence the reticulocyte count. Looking at the literature, we found that the reticulocyte cells are targeted by a type of malaria, which is the Plasmodium vivax. This type starts in Africa, but is less common now than on other continents such as Asia and South America. This suggests that the African population developed a resistance to this type of malaria. There is many hypotheses, and we wonder if the A5 and A43 could be associated with this resistance. This will be a story for another time since we still don't know the answer. In conclusion, we detected positive and balancing selection in the CYP3A cluster and balancing selection in the CYP4F cluster. Our pipeline allows us to detect coevolution and co-regulation between genes in a cluster, and we also saw difference between ethnicity, which could potentially suggest a difference in the metabolization of drugs between ethnicity. This also highlights the importance of considering ethnic diversity in biomedical research, which are enriched in European descent. The next steps of our study is to investigate and understand the evolutionary origin of our result around the A5 and A43. In our study, we mostly focus on the gene, but focusing on the pseudogenes can also give us great insight of how the genes in cluster evolve. Lastly, we will want to look at other CYP which are also involved in cluster and see how they can co-evolve together. I would like to thank the laboratory, the subventionary organism, and you for your attention.